Hello, everyone. Sorry about the technical difficulties. I'll keep this short. Welcome to VanBug. Thank you to Big Research Day for hosting us, and thank you to our sponsors. Our intro speaker is a PhD candidate in Lindsay Etlis's lab, and he'll be talking about using soil microbes to sustainably produce high-value lipids. Raphael?
Thank you. Thank you very much for the invitation. And I apologize, this is not going to be a bioinformatics heavy talk, but I'll promise that I'll do mention some bioinformatics. So this might be a bit not a common talk for this audience. Well, I like to start with the big picture. So the future. Is it going to be bright and green? Well, we do face some serious problems. We're dependent on petroleum, and we have a growing world population. What that means is that is global warming and increasing demands in energy, food, and products, which is threatening our future by more pollution and destruction of ecosystems. But there's hope. We're on our way to make better, greener technologies, and I'd like to talk about this as the agricultural revolution. It means moving away from these bad pictures, using petroleum as a resource, and shifting towards a bio-based economy, using more sustainable methods. And in the future, we'll rethink what, where, and how we grow agricultural products that get empowered by biotechnology. So, so this will be fueled by the development of a bio-based economy. So we have the opportunity to use agricultural waste or high-yield bioenergy crops to produce energy, to make fuels, chemicals, build materials. And we can even reinvent the way how we make food, such as cooking oils, and can transform products like lignocellulosic products that are inedible into consumable ones. So for example, here you can see agricultural waste products like wheat straw or corn stover, and bioenergy crops like silvergrass or switchgrass, which we can use to make green bio-based products. So how will we get there? Bioprocessing technology allows us to transform plant biomass, composed mainly out of cellulose, hemicellulose, and lignin, into sugars and aromatics. We can use physical treatments and chemical and enzyme treatments to convert this biomass into these sugars, and then use fermentation technologies with microorganisms to convert sugars into products. And the industry mainly focused on making biofuels traditionally. However, we do to empower this agricultural revolution, we need other methods and develop other strains and biotechnological solutions to transform biomass into products like bulk chemicals or fine chemicals and materials and food. So here comes this soil bacterium into play. I'm working with Rhodococcus jostil RHA1, or just called RHA1. Which can accumulate high amounts of lipids. It can grow on a variety of substrates. It can be cultivated with high amounts of feedstock input and is amenable to genetic manipulation. And here you can see on a picture, electron microscopy image of an RHA1 cell filled with lipid vesicles. This is a really cool feature of this strain, and we are interested in studying and exploiting the lipid production in RHA1. So the main lipids in RHA1 that are produced are triacylglycerides or tags, This is a glycerol transesterified with 3 fatty acids. And this pathway is catalyzed by 4 different enzyme classes, which are encoded by many seemingly redundant genes. So for example, we find 16 homologs of these ATF genes, these acyltransferases, coding for the last step of the TAC production in RG1. So we're interested in identifying the key components for the synthesis of TACs or other lipids. And here comes a little bit bioinformatics into play. We did an RNA-seq experiment and used different conditions to identify dysregulated genes in RHA1. The one condition we used is nitrogen limiting, which induces lipid accumulation in the stationary growth phase of the cell. So we were able to identify dysregulated genes and show their operon organization. And that gave us a list of key candidates which we are investigating further. So to do this, we looked more closely at some interesting candidates that were mainly overproduced when lipid accumulation occurs in the cell. And we wanted to pin down the exact time point and did RT-qPCR experiments on time-resolved manner to show the involvement of certain ATF gene candidates. And indeed, we see, so C- here indicates carbon-limiting condition, which where the cell doesn't produce lipids in stationary phase, and N- indicates nitrogen-limiting conditions where the cell do accumulate lipids in stationary phase, and the dashed lines show the time point when carbon is either depleted or nitrogen is depleted, and when nitrogen is depleted, the cell will make lipids, and this and the activation of some ATF gene candidates correlates with the production of lipids. So we have we focus mainly on the ATF8 gene shown on the bottom left, and the ATF10 genes shown on the bottom right, which are highly transcribed under nitrogen limiting conditions. And we were able to further confirm this by creating knockout mutants, so deleting the ATF8 and the ATF10 gene, and also creating a double knockout mutant, deleting both genes in a strain, to show the reduction of lipid accumulation and the involvement of these 2 genes in the TAC synthesis pathway. So these are just a couple of examples, and we're focusing on identifying and characterizing the role of other genes involved in this pathway. And I actually also wanted to talk a bit more about exploiting or engineering RHA1 for the production of high-value lipids. So TACs, triacylglycerides, are usually not of high value. So the goal is to engineer a strain that can make lipids that are more useful for the industry. And here we are developing a technology in the lab by rewiring one of the lipid accumulation pathways in RHA1. And we actually want to commercialize this technology, and we want to file a patent for this. So, and we haven't filed it one yet, so I cannot disclose too much of it. But what we are then proposing is that we can make lipids in RHA1 which have unique properties, are more cost-effective, and for the use in certain industry sectors, and are more sustainable and ethical. And together with a friend, I created Rhyme Biotechnology, which is aiming to commercialize this. And another really interesting aspect of RT1 is that it's able to use also aromatic compounds that come from lignocellulosic biomass. And I've briefly talked about biomass being comprised out of cellulose, hemicellulose, and lignin. And lignin is the other major carbon source in biomass, and it's made out of these aromatic compounds, which the industry right now doesn't have a lot of usage for. And another goal of my thesis is to identify pathways in RT1 that can use these aromatic compounds, and to kind of in a biological funneling process use a variety of aromatics to lead them into the central metabolism to make these value-added products as another feedstock resource. And I've done an RNA-seq experiment where to identify pathways that are activated under these conditions. But this is still ongoing work. With this, I'd like to thank my lab, my PI, Lindsay Altis, all the past and current members, especially Carlos Diaz and James Round, and my trainee program, Ecoscope, and all the funding agencies. And thank you for your attention. Happy to take any questions. If we have time. Yeah. So we have identified a gene that can produce a compound that is very valuable for some industries, for example, cosmetic industry, and we're patenting this process of overexpressing that gene to make this compound. Does that answer your question? Okay. This feature can be found in a lot of bacteria or other microorganisms, and even eukaryotic cells that under some stress conditions, for example, nutrient stress, in this case nitrogen limitation, they accumulate lipids. And so one reason is that it is for carbon storage, very likely. So because there's an overload of carbon, and then the cell tries to store that carbon for times when there's more nitrogen available, and then reuse it to grow again.
Thanks again.
Thank you.